Hello, everyone. This is Derya Unutmaz. I'm the principal investigator and director of JAX Laboratory Center for MECFS. I'd like to tell you about the work that we've been doing for the first couple of years. This is going to be a very big summary, so bear with me. So first of all, let me give an overview of our center. So we've set up a collaboration with Bateman Horne Center in Utah, where we collected clinical samples from more than 100 MECFS patients over a period of 4 years. We've acquired 3 time points from most of these patients, as well as a control group that is age and sex matched. These are blood samples as well as fecal samples. The idea was to perform a very extensive immune profiling, which I will tell you about, metabolomics analysis, and also microbiome profiling to determine what is the differences between both the healthy and the patient groups, but also trying to link these data together to come out with a computational interactome that will allow us to identify new mechanisms and new hypotheses that we can eventually test to understand both the pathogenesis of the disease as well as create several biomarkers. So just to give you a little bit of background about the immune system, which is our main area of interest, you can roughly divide the immune system into 2 parts. You have the innate immunity and adaptive immunity. Innate immunity is really the first responders, your epithelial barriers, your macrophages and natural killer cells and complement and so on. So these try to stop bacteria or viruses or any other threat, but they don't really know what type of bacteria or virus that is. In fact, if you're infected with SARS-CoV-2, this is the first line of defense. There's an interferon response, and that really slows down the virus. But the real immunity is established by adaptive immunity, which consists of B cells and T cells. B cells, as I'm sure all of you know, produce antibodies that you've been hearing about in the past couple of years because of COVID. These antibodies will lock onto surface of viruses or bacteria, and they will neutralize them and prevent them from entering into the cells or destroy them. And you also have T cells, and T cells are a bit more complicated because you can imagine them as sort of the commanders of the immune system or the regulators. They have subsets. There are probably dozens of them. You have cells that we have effector cells that can identify cells infected with viruses. They have a cytotoxic or killing ability. They then recognize and destroy these infected cells. You have other cells that give help to B cells and other parts of the immunity, the innate immunity as well. So this is a very coordinated effort that prevents infections from happening. But of course, this is a very dangerous system at the same time, so it has to be regulated very, very tightly. It's kind of like an army, and if the regulation is not done properly, the immune system itself becomes the problem. Again, we see this a huge issue with COVID. A lot of people who have severe disease actually probably or due to this excessive immune response that has to be controlled by suppressing it. So again, just to give you an idea of the complexity of immune subsets, in particular T cell subsets, there's a complex labor of division here. You have cells that we call conventional T cells that are divided based on their effector functions. For example, the one I mentioned, cytotoxic cells, kill virally infected cells that can be immobilized against tumors or cancer as well. You have what's so-called Th17 cells that we're very interested. These are actually respond to bacteria and fungi, but they seem to also regulate or interact with the microbiome a lot. They're affected by the microbiome, and they can cause autoimmunity or chronic inflammation. So again, these cells can be dangerous. That has to be regulated. Then you have other type of cells, Th1, Th2 cells, that respond to parasites and bacteria and viruses, depending on the threat that you have. But again, they have the downside of causing allergies, asthma, or autoimmunity. You have cells that help B cells to produce antibodies like T follicular helper cells, and then you have cells that regulate all these responses called regulatory T cells, which are also very important because if you don't regulate them, then you get these immune diseases. We also have unconventional T cells. In particular, we study mucosa-associated invariant T cells that have a restricted repertoire of recognition. So they don't recognize everything like these T cells do, but they can recognize, for example, bacteria or bacteria-infected cells. I'll tell you a little bit more about this. They recognize a metabolite derived from vitamin pathway produced by bacteria, actually, and we think also they regulate the microbiota. You have natural killer T cells and gamma delta cells and so on. So the complexity doesn't end there. So you also have flavors of these T cells that can have different effector functions, subsets, let's say, but they also encode, they also express these so-called chemokine receptors such as CCR4, CXCR5, CCR4, etc. And these allow these T cells to go into different locations. So you don't want cells that are destined to go to gut to go into lung or vice versa. So these are very important, kind of like address codes that tell the T cells to go where they're needed, or where they can be resident or localized to. Any dysregulation there can also cause major problems. So keeping that in mind, we've set up to perform this extensive profiling to determine the frequencies as well as functionality of these subsets that I just mentioned, including B cells and other innate subsets. So we have 150 patients, age 6 match controls, 3 different time points that were collected on a yearly basis. And these patient cohorts were divided into 2 groups, and we think this is important for our cohorts because we have a subgroup that about half of the patients have the disease less than 4 years, and the other had the disease for more than 10 years. And this is, I think, quite unique to our study as well. We aim to identify frequencies in functional subset of immune cells, as I mentioned, and generate about 300 immune cell function data points per patient. So this generates a lot of data, and we think it will probably be the largest prospective immune profiling data in depth and breadth available for MECFS patients. We've also now included 40, actually it's now 35 long COVID patients that will be followed in at least 3 time points. We've actually acquired blood samples for the first time points and some of them in the second time points. As you probably know, long COVID has some overlapping symptoms with MECFS, so this is of great interest to see how that compares to MECFS immune or other dysfunctions or metabolic dysfunction, for example. We think that this might be both helpful for the long COVID patients, but also an opportunity to understand what really is going on in chronic fatigue syndrome. The data that we generate, as I mentioned, is very extensive. Actually, we have done a similar study in a previous study. This was a cross-sectional single time point study of about 200 MECFS patients, and we've identified using some machine learning approaches as well a set of immune markers that were very, very significantly different between healthy controls and MECFS. And by combining these different markers, again, using a random forest analysis, a machine learning analysis, we could come up with a set of biomarkers that would identify the patients, or that would discriminate the patients from the healthy controls up to about 90% or over 90% accuracy. This was quite remarkable, suggesting that not only there was major differences in the immune system, but these would perhaps could be utilized as potential biomarkers in identifying the MECFS patients. So the other topic that we're very interested in is the microbes within us. In fact, there are trillions of bacteria that live in our gut, in our skin, and other mucosal areas. There are probably more bacteria in us than cells, so it's unclear whether we're there for them or they're here for us. But they certainly help us a lot in terms of digesting our food and producing many different metabolites that actually affect our physiology, in particular the immune system, but also nervous system and other physiologies are affected by these metabolites. And we're just beginning to understand the enormous impact of these microbes within us. And one aspect of this microbiome, so-called, the sort of the universe of the microbes within us is also personalized. There are many different strains of bacteria, and their distribution can be very, very different from person 1 to person 2 to person 3. Even identical twins living in similar areas might have different microbiome compositions. And this, we think, also, as I said, impacts the immune system and therefore creates a different immune profile as well for each person. So this is a very challenging problem. We have to come up with both a personalized version of this, but also identify what are sort of the good guys and the bad guys and how they impact or change the balance for disease. And this microbiota is actually pretty dynamic. There are resident bacteria, resident in the sense that they have colonized there, and most of them don't change for a while unless you have something happening. If you change your diet, if you take antibiotics, if you have some pathogenic infection, immunodeficiency, chronic disease, aging turns out to be a major factor, then you can have these communities change. So they are quite dynamic in that regard, and also this allows us to manipulate them and provide an actionable type of information that could reconstitute the disturbed balances of microbiome and in extension for the immune system and other physiological parts of our body. So as I mentioned, there is an equilibrium of this microbial ecosystem. So you have those that we call symbionts that are usually very helpful for us, that we really need them. You have commensals that are sort of in the middle, and then you have what's called pathobionts, which are in a steady state, they don't cause problems, but something might happen and tip them over, and they can cause chronic inflammation. And this could cause a lot of other issues, and they've been shown to be involved in many chronic diseases, including autoimmunity, even Alzheimer's and chronic heart diseases and many functions of immune system. And even this balance, probably this ecosystem is involved in responsive immune system to cancer. And we think that it's also a major contributor to MECFS, which we have some good evidence for. So the project design for the microbiome is again very similar in the sense that we collect stool samples from patients that I just described. We provide a sort of a taxonomic profile, which includes sequencing of these bacteria, so we can identify what type of strains and what sort of frequencies they're in. But we're also had designed a sort of a functional assay where we can address whether bacterial produced metabolites or products can affect the immune system. So this is a sort of a functional screen, as well as looking at the blood to determine what proportion of the metabolites in the blood are derived from microbes versus the host. So actually we've had some very interesting results, which was submitted recently, which were actually somewhat similar to Ian Lipkin's group in Colombia. We've actually co-submitted these papers, and they're under revision right now. So the major points is that we have in the short-term patients, the ones that had the disease for less than 4 years, there was a significant microbial dysbiosis. Dysbiosis is the one that I mentioned, that balance of the good guys and the bad guys is disturbed in advantage of the bad guys. Long-term patients, the ones who had the disease for more than 10 years, sort of reestablish a healthy control look-alike, but they also have a major metabolic dysbiosis, probably leaking from the gut microbiota. This could be life changes, GI disturbances, and various other factors. We're trying to figure that out. So again, this is sort of our framework goal for the host microbiome interactions to decode the metagenomics part of it, meaning that we want to identify different strains of bacteria. This is also a very big universe. Actually, a lot of this information has not been yet understood, and we're trying to provide knowledge sets there as well, identify the metabolites, and then link this to the immune dysfunction that we might find. So we think that this complex interactome type of analysis will provide us a sort of a host phenotype that could give us a better picture of what is going wrong in the MECFS patients. Again, these are all very much linked, and as I mentioned, there's a huge link of the microbiota to immune dysfunction. It is quite well established now that the changes in certain big phylogenetic ecosystem of bacteria, such as Firmicutes versus Bacteroides, if these proportions change, they can cause autoimmune diseases. They're involved in type 1 diabetes and multiple sclerosis and arthritis. Again, this is an indirect effect, obviously, because the bacterial dysfunction or dysbiosis affects the immune cells, in particular Th17 cells, the mucosa-associated invariant cells that I will mention in a second, and dysfunction in the regulation of the immune cells as well. So it's reasonable to assume that something similar might be happening in MECFS. And one set of cells that I already mentioned, which we're very interested in, is called the MAID cells, or mucosa-associated invariant T cells. These are, as I mentioned, are non-conventional T cells because they recognize metabolites produced by vitamin B2 pathway, which is also produced by bacteria, and they recognize this with a molecule called MR1. So they have a restricted recognition pattern, but we think that they are involved in regulating or sensing the changes in the microbiota. So if there is a dysbiosis, probably these MAID cells are going to get activated, and they will provide communication information to other immune cells as well. So we think they could be both biomarkers as well as provide us some knowledge about what can be put back to normal or back to the steady state. Finally, our last set of analysis involves metabolomics. This is again another very huge system-wide analysis. So the idea was to perform metabolites, to analyze metabolites in blood samples. And we're doing this in collaboration with a company called Metabolon. They have a selection of about 1,000 metabolites. These are very highly well-established assays. This is a sort of a gold standard company which does this, and I think Cornell Group also uses Metabolon for their analysis, so that allows us to compare data sets as well. So we have already accumulated about 500,000 data points. We've done this all time points for all patients and healthy controls. And we're already analyzing, and we have the first set of data that I mentioned is already submitted. But of course, the key that we are working on right now is to link this to the immune and microbiome perturbations, which what we will do in the remaining time for these centers. We've already identified some interesting perturbances in the metabolites that potentially link the microbiome to the immune system, such as short-chain fatty acids like butyrate, indoles that are involved in tryptophan pathway, bile acids. So these are important because there's information, there's knowledge that these metabolites regulate the immune system, and they seem to be perturbed in some of these patients. So again, it allows us to establish new hypotheses or paths to assess whether they're directly involved in the MECFS pathology, and if so, then they can be interfered, and we can they're sort of actionable knowledge because we can try to fix them. So we're also, as I mentioned, trying to perform this functional assays, which we call culturomics. This is a little bit of a complex set of assays, so I won't go into details of it, but basically the idea is that we are growing individual bacteria derived from patients and matched controls, and we've generated thousands of these bacterial strains and collect their supernatants and add them to immune assays where we test whether they have particular function on particular immune assays. And we've generated lots of data on that. We've already identified some very interesting bacteria strains that affect what's called the AHR pathway, oral hydrocarbon receptor pathway. Again, I won't go into details of it, but this pathway is also involved in a variety of immunological regulation. So the next step is really the multi-system analysis. So we're in the past 4 years, we've been accumulating an enormous set of data thanks to incredible support from Bateman Horne Center and the patients who've participated in this analysis, in this research project. We've collected, as I mentioned, 150 patients over 3 years, and including the healthy control. So we've have an enormous amount of data. We're now finalizing the immune profiling analysis, which we had a little bit of a setback because of COVID, but we're just catching up with that as well. We have completed all of our recruitment, so we have all the samples, most of the metabolomics data as well as the microbiome data. So the remaining time that starting this spring, hopefully we will be starting to put this together and trying to do lots of data analysis and machine learning analysis for this integrative analysis. So as I said, the idea is twofold, so both to try to create some biomarkers, but also importantly, come up with the mechanistic hypothesis that can be actionable, that can provide the path for clinical trials to try to interfere with them or to return them back to normal states, both in microbiota and metabolomics. I think these are going to be quite feasible. So that's really an exciting aspect of our study. And lastly, I mentioned about long COVID. We have about 35 subjects already recruited. We've required second time points. We're requiring the third time points as well. And we're going to be doing both the metabolomics and immune profiling. This was a sort of an extra study that we came up with because of COVID, of course. And we think that these long COVID patients have, not all of them, but some of them have MECFS-like symptoms, and they will allow us to both understand MECFS, but unfortunately, this will also create millions of people with MECFS-like symptoms, so this is of great importance for us to determine. One thing that's unique in this case is that if this is caused by SARS-CoV-2 infection, which very likely it is, then we are actually identifying the initiation of MECFS-like symptoms, because this has been missing in the field. Most patients who had MECFS in the research had for many years. We do have this unique cohort of patients that have less than 4 years, but we don't exactly know how or when it started or what was the trigger factor. So here we know exactly the trigger, we know the virus, and we can follow them. And these subjects will be important to be followed for the next several years as well. And I think this also greatly increased the interest to the field, which is great news for MECFS patients, and we think that we can contribute based on the data that we've already accumulated and trying to understand both long COVID and MECFS. So I'd like to acknowledge our wonderful team in this center. I run on a human immunology group. We're focused completely on analyzing the immune system. Of course, we have a wonderful MECFS clinical team. None of this would be possible without them, Cindy Bateman and Suzanne Vernon. They've been collecting in an incredibly meticulous manner, recruiting the patients and collecting the samples. The quality of these samples are very, very important, because if you don't get good quality, you will not get good data. Our microbiome lead and associate director is Julia O. She's a microbiome star in the field. She's already generated a lot of data that some of it is already submitted. Zudang Yao at UConn, he is an expert in metabolites, bacterial metabolites. He's a biochemist, identifying some new metabolites that from the bacteria that we isolate. Shuzhao Li, who recently joined Jackson Laboratory from Emory University, he's one of the lead experts in the world about metabolomics. So he's helping us with data analysis of this massive data generated that I mentioned. And Du Yuchar is a computational biologist. She's been working on aging field, but will help us with this integrative approach that I mentioned, trying to put everything together. So I think that's it. Thank you very much.
Thank you for joining us today. My name is Jessie Brown-Clark. I am an advocacy manager at SolveME and a member of the Community Advisory Committee for the NIH-MECFS Research Network. Today, I am speaking with the group at Jackson Laboratories, a part of the NIH Collaborative Research Center. I'm going to kick it to them to introduce themselves and their roles on their team.
So I'll just start. I am the director of the MECFS Center at Jackson Laboratory. I am a professor there and human immunologist. I've been working on T cells for the past 30 years. And we've been trying to understand both the T cell biology and in general, human immunology in the context of different diseases. Julia.
And I'm Julia O. I'm Associate Director of the center here and Associate Professor here at the Jackson Laboratory. And my group's expertise is in computational experimental models of human microbiome interactions. And so here we've been trying to understand how changes in the microbiome might be interacting or begetting changes, for example, in the immune system that's occurring in these patients.
Suzanne.
Hi, I'm Suzanne Vernon. I'm the research director at the Bateman-Horne Center. The Bateman-Horne Center is the clinical core for the JAX CRC, and our role has been to recruit and characterize patients, MECFS patients, long COVID patients, and healthy controls, and collect samples and send the samples to JAX Lab for analysis.
Great, and thank you so much for joining us today. Your presentation and your work is so interesting, and it generated a lot of thoughts and a lot of questions. So these questions I'm about to ask were posed by the entire Community Advisory Committee. So I will go ahead and get started. So the first one that we have is to really set the stage for your presentation. Could you please provide a brief overview explanation of what T cells do in the body and explain the innate immunity and adaptive immunity, what that is?
So I'll take that question because that's right on my alley. And first of all, thank you very much, Jessie and MECFS and Alice and other organizers for this meeting. I think the questions were great, so hopefully we'll be able to respond to them. So let me just start with first of all, what's the difference between innate and adaptive immunity? Of course, the immune system is very complicated, has many parts. It's sort of like an army. You know, you have the Navy, the Air Force, and the land forces, and many subgroups as well, because the immune system responds to threats, and it doesn't know what kind of threat it's going to be exposed to. As we have experienced in the past 2 years, COVID has been a tremendous threat for our immune system, and it had difficulty in recognizing it. But it's also prepared for these unknown or new threats or dangers or pathogens. The first layer of this defense system is the innate layer. So the innate immune system actually recognizes certain patterns of viruses, bacteria, or parasites that could be, for example, the DNA of the bacteria, or it could be the RNA of SARS-CoV-2 virus. So it looks at these patterns, and it can tell that this is a virus. It says RNA virus, but it wouldn't be able to tell whether it's influenza or COVID virus or something else. But it would know that that's a threat. And it usually works at the mucosal layers, when the pathogens are trying to infiltrate us, and it tries to slow things down. That's its first purpose. It's really sort of bring the soldiers to the border so they can start defending, even though it doesn't know exactly what type of pathogen. But it has a second and very important other role. And actually innate immunity starts within hours. So it really responds very quickly. And we know this, for example, from SARS-CoV-2 infection, a number of people who are exposed to this virus never even get sick because the innate immunity is able to slow down or really stop the virus at these borders so that the second layer, which is the adaptive layer, has time to be trained. And actually, the second role of innate immunity is to tell or go to the lymph nodes and bring the message that there is an enemy at our border. We're not sure what kind of enemy it is. We think it's a virus. We think it's an RNA virus. Start building your factories that will produce these smart missiles, which would be antibodies, and start training your soldiers specifically to recognize that virus. And that's exactly also happens during COVID, but that takes at least a week or 10 days. So you need time to get to that point. And that's the reason why the vaccines also work, because what the vaccines does is that it trains the adaptive immunity specific to that virus, and not only the virus, but also a piece of the virus. For example, you want an antibody that will bind to the spike protein of SARS-CoV-2 virus so that you can neutralize it. And those antibodies have to be made from scratch, and the factories have to be built as plasma cells and so on. So it's sort of, they work in a very collaborative way. And then once the adaptive immunity starts, the T cells start to grow. They also go and give commands to the innate immunity. So that brings me to the T cell biology a little bit. What are T cells? So T cells are part of this adaptive immunity. So they are both the commanders of the immune system, but also they can be the snipers or the special forces of the immune system. I like making these analogies because I think it makes it a little bit more clear. In fact, they work exactly like that. For example, the CD4 positive T cells, they really send commands in terms of cytokines, in terms of chemokines. They tell the innate cells where to be. They help the B cells to make the antibodies, so they are very critical on that. They recognize very small pieces of the antigens called peptides, so they don't really know the structures, but they know that this is specific to that virus or that bacteria and so on. And then they also have a regulatory function because immune system is dangerous and it can lead to chronic conditions, and we think it even is responsible in MECFS. So it has to be controlled very precisely. Even in COVID, a lot of people die because of overreaction of immune system. And in fact, in the late stages, you have to use steroids to suppress the immune system so that it doesn't kill you, trying to kill the pathogen. And then we have these cells called regulatory T cells or Tregs that actually are sort of like the bureaucrats, and they say, okay, that's enough. You need to slow down now. Stop shooting around. If they don't function, then you get autoimmunity and all kinds of chronic inflammation. Then you also have these sort of the snipers or special forces, whatever you like to call them, the cytotoxic T cells, which will go and seek out these infected cells with the virus or sometimes intracellular bacteria, and then they will destroy them. They will literally kill the cells. And the reason is that if you cannot stop the virus before it enters into a cell, you cannot neutralize it with your antibodies or smart missiles, then you have to kill the cell so that the virus cannot replicate in the cell and spread out. And these are done mostly by CD8 positive cytotoxic cells, which really act as assassins or snipers. They're very specific. They will only kill those that are infected with the viruses. Of course, again, they have to be controlled because if they start killing too many of your cells, then you're in trouble. That can cause immunopathology. Or if the immune system thinks that there is a danger, but there is really no danger. And we think that, that's part of the problem maybe in MECFS, at least that's our hypothesis, that the immune system is constantly fighting a war that is not present anymore. It thinks that there is a virus or a bacteria, and it's doing some damage to the system without really beating anybody. Of course, it's very complicated. We'll talk about the microbiome, metabolism, and all those interactions. This is a sort of very, very superficial way to explain a very complex army within us.
Great, thank you so much for that. That's so interesting. Yeah, and so our next question is, you design your studies with a disease duration of under 4 years and beyond 10 years. Can you explain how and why you chose those delineations?
I think this question is best for Suzanne to answer, because they're our clinical partners, and they are the ones who actually designed. So Suzanne, it's yours.
Thank you, Derya. So when we designed the study, it was based on the fact that we had actually worked with JAX and Derya before on an R01, where we generated some immune profiling data, where actually Derya generated some immune profiling data that showed some very interesting immune profiles on the blood samples from MECFS patients who were mostly greater than 10 years in duration of illness. And in fact, many of the studies that have been published to date, looking at immunology, immune system of MECFS, were on patients that were mostly longer duration illness. And that's just kind of the nature of the beast, actually, because many of our patients take a long time to get a diagnosis. So by the time they're enrolled in research, they're ill for a long time. So we saw this as an opportunity, the JAX CRC, as the clinical core, to enroll patients that were of shorter duration illness. There were a few studies in the literature, including some studies done by Columbia and also in Australia, showing that the shorter duration illness MECFS patients looked potentially a little bit different. They had different immune profiles. So we decided to try to aim to recruit shorter duration illness. So the cutoff of less than 4 years is somewhat arbitrary, but the work from Columbia showed that patients less than 3 years had a specific type of cytokine profile. So we initially aimed at less than 3 years, and we were finding that we were struggling with kind of a 3-4 year cutoff. So we in council or in discussions with Derya, we moved that up to less than 4 years. And then, so we recruited 75 patients less than 4 years and 75 patients greater than 10 years. And the really interesting thing about this particular cohort is that we not only have one sample, we have actually longitudinal samples. So each of the people involved in this study have multiple blood and microbiome samples collected over time. So the results from this, I think, are going to be one of a kind, very unique. It's one of the larger data sets of MECFS patients, and it's one of the few data sets that actually has longitudinal sampling.
Wow, thank you so much. So the next one is about immune profiling and identifying immune signatures. So you state that using machine learning approaches, you identified a set of immune markers that were very significantly different between MECFS and healthy controls, that they can predict within 90% accuracy. What would it take to make that a real biomarker that could be used in clinical care?
Thank you for that question. The study that's been referred to is what Suzanne also mentioned. This was our first work with MECFS funded by NIH through an Aero grant, where we indeed found remarkable differences between the MECFS and the healthy controls, which are who are obviously matched for sex and age. We found some other very interesting things. For example, age did matter. For example, people over the age of 50 had a different sort of biomarker set or immune profile than those who had less than 50. It almost seemed like there was a little bit of an accelerated aging of the immune system in the patients. And in fact, we've done a very extensive immune profiling. When I say immune profiling, what we're doing is what I mentioned earlier. This army is very complex. So you have these T cells, and you have subsets of T cells, and those that are regulatory, and then they regulate different cell types, and so on and so forth. And they also have different functions. So we analyze both the frequency of these very in-depth analysis of these subsets, but also their functionality. And it turned out that the functional analysis was even more valuable. When I say functional, for example, we elicit the cells to produce what they would normally do. For example, they're making a specific cytokine called, let's say, IL-17. What IL-17 does is that it talks to the mucosal cells. It tells them what to do, prevents the microbiota from passing through, and has many other functions. So there's a specific subset of cells that will only make IL-17. And when there are many other examples. So we found that specifically those populations functionally were defective, or I shouldn't say defective, but they were abnormal or perturbed. So when we combined all this data, and we had several hundred data from each patient, and it was over 150 patients, and compared to healthy control, I think at the time it was also the largest immune profiling done in patients. We applied some machine learning analysis because one human cannot really make sense of so much data. And using this machine learning called random forest analysis, we were able to come up with the top 10 markers. Together, they're able to tell who's a patient, who's not, up to about 90% specificity and sensitivity, which was quite remarkable. It's remarkable because the immune system is very, very different from person to person. Even identical twins can have completely different immune profiles because your immune system is shaped by the environment, the viruses, the bacteria, all these things that you're exposed to, your microbiome, the stuff that you eat, your metabolites. But regardless of that, we were able to see this change in this population, and that of course really excited us. In fact, we asked exactly the same question that was asked in this question. How do we turn that into a sort of a biomarker that could be, that could both explain the disease and create some targets for therapy, but also can we better diagnose MECFS patients? I mean, this was 5-6 years ago, and it's interesting because at the time, a lot of people were still skeptical that MECFS is a real disease. I was not obviously one of them. And to see this biological data was very satisfying in some regard because, clearly something was terribly wrong in these patients. But you wouldn't be able to pick that up with, standard laboratory tests. They could all be very normal or, not different than some other chronic disease. So then exactly we asked that question because of this problem that the immune system is very diverse and it can change over time, over years in age. It does make it difficult to just use that data as a clinical biomarker, because then you have to look at more people, and you have to do this expensive flow cytometry and so on. But we said, if we can find this difference at the immune profile level, we should be able to find what causes that, for example, the microbiome or the metabolites. And perhaps by combining those, we might be able to come up with a much simpler and much accurate clinical biomarker. And that's why we applied for the center, actually. I mean, that was one of our key questions, and we're hoping that we're getting close to that. But it was it was really revealing, even for me, to see those profound changes.
Wow, that's fascinating. Great. So now we're going to move over to talking about the microbiome. So you state that in short-term disease duration patients, you see significant microbial dysbiosis, and in long-term patients, so more than 10 years, that they reestablish microbial balance, but they show major metabolic dysbiosis. And you say probably leaking from the gut microbiota. Is this what is referred to as leaky gut? Or are you saying that microbial dysbiosis in early stages somehow leads to metabolic dysbiosis? Can you please further explain this?
Yeah, of course. And thanks so much for the question. So let me first define microbial dysbiosis, as it's a fairly broad term. So in its simplest form, it means changes, usually for the worse, in the overall microbial community, like changes in its overall diversity, which is the number of microbes present, or the presence or absence, the lack of specific important commensal microbes, or an enrichment of potential pro-disease microbes. And what we saw, indeed, in the short-term patients is that these patients have much lower microbial diversity, and they have fewer specific microbes that are beneficial and that produce what are called short-chain fatty acids, specifically one called butyrate, which are these chemicals which have important signaling and immunomodulatory roles. But surprisingly, which you alluded to, so these differences are to some degree normalized in the longer term patients, although on the flip side, they have more severe metabolic dysbiosis or changes in these chemicals and a broader panel of chemicals that are kind of floating around in their blood. So why could this be? So we speculated that the early microbial dysbiosis observed in the short-term disease patients could result from a few different factors. So first, the short-term patients tend to suffer more gastrointestinal disturbances, and we know that gut microbiome changes may occur reflecting kind of the different environmental changes that are occurring that are associated with things like the diarrhea, with the other GI disturbances. So second, it's possible that patients might try a different range of interventions that impact their gut microbiome. We know that the gut microbiome is dynamic and influenced by numerous intrinsic and extrinsic factors, including age, including diet, including probiotic use or different medication use. And these can dramatically shift gut microbial community composition as well. And so, what we hypothesize is that, there's these different factors that are ultimately leading to this microbial dysbiosis in the short term. And while these are primarily observed in the short term, they have cumulative and longer term effects where, you might have damage that's caused by an initial trigger, but then results in cascading events. So, for example, MECFS progression might begin with the loss of these beneficial microbes, those short-chain fatty acid producers. That in turn can affect gut epithelial barrier integrity and then the immune response, and then result in more pervasive gastrointestinal phenotypes that's later reflected in plasma metabolite levels in the longer-term patients. So I do want to have a caveat here that these are hypotheses and association studies. These are the microbes associated with the short versus the long-term patients, and that subsequent studies are needed to prove mechanism.
Thank you. Now, is this the reason why people with MECFS are likely to have other diseases as well? And could this be a reason that we see MECFS comorbidities develop over time?
So certainly, we believe that these differences can certainly have farther reaching effects. And so, again, taking up that example of the depletion of butyrate producers in the gut, which also is associated with reduced plasma butyrate levels, so the levels in the blood, and again, emphasizing that this is an important immunomodulatory metabolite. So this is, we don't know necessarily that this is causal, to what degree the plasma butyrate levels are dependent on the actual production by the gut microbiota. But when we do know that when there's less of this immunomodulatory chemical, less of other potential beneficial immunomodulatory organisms, taken together with the immune dysbiosis that Derya's observed in the blood, and we hypothesize that the microbiome can prime or sustain an aberrant immune response following disease onset, likely resulting in a range of the comorbidities given the dangers of that aberrant immune activation.
Wow, that's fascinating. Thank you so much. So now we're going to move over to talking about metabolomics. Can you give us a brief explanation of metabolites and why they're important to your research?
So I'll try to answer that. In fact, I'm really happy that we metabolomics or metabolism was part of our initial grant proposal, because it turned out to be very, very exciting. So the reason why we thought the metabolites or metabolism would be very important is because, as I mentioned earlier, when we observed this immune perturbation, we had to ask the question, why is this happening? It could be that the immune system is, there's some sort of a trigger or some sort of a irritation that it's responding to, or it could be that it's sort of the steady state hemostatic level is perturbed, or it kind of falls off the cliff, and that in turn perturbs the microbiome, as Julia mentioned, to where we know about 30% of our metabolites are derived from. These bacteria make these metabolites, and in turn, those disturbances in the metabolism could act on the immune system, which would increase this cascade of events, sort of a vicious cycle that would make things worse, or it would prevent from immune system recovering, going back to the cliff. So that was our kind of hypothesis. And the other thing that we thought metabolites would be very interesting is because there's a huge number of metabolites that are never analyzed, and we don't even know what they do. The number of things that we analyze in the lab as diagnostics is not even the tip of the iceberg. So, we've partnered with this company, Metabolon, who actually does this stuff, and we collected plasma samples, from blood, and we said, what sort of metabolites do we see, and what are the differences? And it turns out that there's about 1,000 metabolites that they can very reproducibly detect in the blood. Probably there are more of them. And these are, when I say metabolites, these are lipids, amino acids, small molecules, because, our biological machinery has a lot of enzymes, a lot of pathways, and each of those things produce some sort of small molecules that either accumulate as a byproduct or they themselves have an effect. Julia mentioned butyrate, which is what's called a short chain fatty acid. And these are produced by bacteria, but they have a very important function on the immune cells. They can either suppress it, or in some cases, they can activate certain cell types. There are many, many examples of these things. So in fact, we were very excited that, we saw, again, with the metabolites, a huge amount of differences in the patients versus the healthy controls. The other aspect of metabolites is that it also allows you to really understand a certain mechanism, or is what is causing the problem in the immune system, or what's causing the problem in the brain, for that matter. Because these are small messengers. In a way, they can go and act on the brain or the intestine or any of the organs. So that's first thing. And then the second is that, going back to an earlier question, it would be relatively simpler, or maybe in future easier, to build a sort of biomarker set from these metabolites, if they can be really quantitative. So in many aspects, this has been a very exciting area. And lastly, I should mention that, by coincidence, all three groups, centers have been interested in metabolism. And so we've all done some sort of metabolism within the context of our projects. And what's really remarkable is that we're finding a lot of similar things. That's also very satisfying because reproducibility has been one of the major problems with MECFS.
Wow. And are you starting to see emerging profiles of phenotypes or other biological driven subsets in the results of your metabolomic analysis?
Yeah, so as Derya mentioned,
Julia, go ahead.
Yeah. Yes, yeah. Derya mentioned earlier, so we use a lot of these machine learning classifiers to try to determine microbes and metabolites that differentiate the patients versus the controls. And what these also allow us to do is to determine what actual features, what specific microbes, what specific metabolites allow us to accurately differentiate the patients. And so using the microbial and the metabolomic data, we could build very accurate classifiers. So with 90% accuracy to differentiate MECFS patients with, as we talked about earlier, the longer term patients having the most significant metabolic dysbiosis. And that's really the most obvious change that we're seeing in the metabolome. And so some of these include those butyrate metabolites that I mentioned earlier. Some other ones are bile acids, benzoate, cholesterol, and other lipids. And these latter has been observed from some of the other MECFS cohorts as well, which I think underscore that we're beginning to now triangulate on conserved metabolic changes, and I think more robust MECFS biomarkers. Again, the reproducibility issue that Derya just mentioned. And so, while the mechanism has yet to be determined for these different metabolites, I think these are really high quality targets to pursue in mechanistic studies. So just as an aside, we're also now begin to, I've talked about the human metabolomes, the chemicals in the blood that are different in the patients, but we're also now beginning to look at the microbe specific. So microbes that have been cultivated from the patients to see if there are actual differences in the production of their immunomodulatory metabolites. And that allows us to begin to see if, these predictions that we've made from our omics data can actually be turned into some hypotheses and mechanisms, because we really think that the metabolites really are the carrier of the signals that mediate the microbial impacts onto the host and its immune system.
Wow, that is so interesting, and thank you for sharing that. And we also had a lot of questions. We're very interested in your multi-system data analysis. What has all of this data analysis taught you about the mechanistic pathway that might be underlying this disease?
So I'll try to respond to that. I don't know if maybe Julia or Suzanne would have anything to add. But first of all, it taught us something that we highly suspected, that MECFS is a very complex disease. It's definitely multifactorial, and that our initial hypothesis that these three different compartments, the immune system, the microbiome, and metabolism, all play a role, very likely in an interactive way that we still don't understand. But that was the goal of the project, and we're still analyzing the data. This is going to take us quite some time, because we have an incredible amount of data generated. Not only from, as Suzanne mentioned earlier, this study is unique in the sense that we have patients less than 4 years, those who had more than 10 years. So we're trying to get into the complexity of the situation where when MECFS becomes more chronic, what things change, and we're already starting to see that, as Julia mentioned, in the microbiome and metabolism. But of course, one of the key questions is, what is the sort of the mechanistic basis of all this? What's going on there? We don't have a good answer yet. We have pieces of it, and then the answer is probably going to be several answers, because we think for some patients it's going to be one piece is coming together, another is something else. There is a common thread, though. The common thread is that clearly in almost all patients we do see this microbial dysbiosis metabolic dysfunction and immune perturbation. Whether one leads to another, and then that sort of causes this cascade is remained to be determined. So we still have some more work to do. Of course, this is not the end. This is just the beginning of starting to make some new hypotheses for future.
Great. And speaking of the future, so what insights do you have now that you perhaps didn't have 6 months ago or a year ago?
So of course, 6 months a year is not a long time in research, but remarkably, this field is advancing very fast, and especially because in the last, I would say, last year, we've been generating. We were coming to the end of the generation of the data, so we're starting to see the outcomes. The most pronounced is the microbiome and the metabolism data that we've completed first, and as Julia pointed out, to see this remarkable dysbiosis and metabolic changes. In fact, there's even changes over time, because that's another aspect of our study, that we have a prospective study, 3 different time points. It's just very both satisfying and very remarkable to see that, because these were all hypotheses, and the data was coming out, and we were actually beginning to see how spot on some of our hypotheses were. And then the other thing was, other centers were also starting to really generate this complete sets of data, and to see that their data is comparable to ours. Of course, there are some differences because their subject populations are different. They're asking some different questions, but by and large, we were starting to see some different things, some similar things. So I think those were the most striking things in the past 6 months to a year.
How exciting! And what are your targets that you think could be worth pursuing in future clinical trials?
Yeah, that's the million-dollar question, and maybe more. I think one of the unique aspects of our research is that we really wanted to identify targets that could be clinically probed or tested, but actually not just targets, but also potential therapeutics. And the potential therapeutics are the metabolites themselves. Because either the metabolites, as Julia pointed out, could come from the bacteria, or the bacteria itself if it's disturbed. So if you can replace it, or if your butyrate levels are too high, if you can sort of bring it down to a level that would be acceptable, and maybe a lot of things will be fixed. So I think there are many potential areas of what I would say mini clinical trials, because no matter what your idea is, when you go to the trial, things may not turn out as you might hope for, but at the same time you learn. I mean, for example, if you test, can we modulate the butyrate levels, and you find that there's not much effect, so you at least know that that's not a key point in the MECFS pathogenesis. Or maybe that if you try it on the patients that are less than 4 years of disease, you start to see improvement, but not in the more chronic patients. So another area that metabolite set of metabolites that we work on is something called Aero Hydrocarbon Receptor ligands, and these are also produced mostly by bacteria, but also our host cells. Aero Hydrocarbon Receptor, or AHR, is a major regulator of many aspects of our biological system, including the immune system. So we're identifying some very specific ligands or molecules from bacteria that binds to this AHR and can signal it and trigger it. So that on itself could be a potential therapeutic molecule in future. And maybe not even just for MECFS, but for other conditions, other chronic conditions, or maybe in autoimmune diseases. And I think that's the exciting aspect of it. And I think looking forward, we hope that the NIH also funds more of these mini clinical trials. Of course, here, Suzanne and Bateman's Horn Center and centers like that become very important, because you have to recruit the right patients and really try these this different potential targets or potential therapeutics.
Wow, thank you so much. And we look forward to hearing more about that in the future, for sure. And so the last set of my questions today are talking about long COVID. So one question that we had was how you are selecting your long COVID cohort. Are you requiring a positive COVID test?
So I'll answer that. Thank you for that question. We decided to include. So originally, obviously, this CRC, the JAX CRC, was started in what, 2017? So 3 years before the pandemic began. And so in 2020, with the pandemic, and then in 2021, as we saw that people with COVID, some people with COVID were not recovering and potentially developing post-COVID sequelae, we kind of jumped on the opportunity to see if we could enroll long COVID patients into our JAX cohort, and to understand, number one, is post-COVID sequelae or long COVID similar to, different from MECFS? What does that look like at the immune, metabolomic, and microbiome level? So we began, we amended our protocol to include long COVID patients in the JAX cohort. And the other thing that's really going to be unique about this particular long COVID cohort is we are also sampling them quarterly. So every 3 months, they come in to BHC and have samples and clinical data collected. And then that information and those samples are sent to JAX for analysis. So it should be super interesting. At BHC, we didn't set super strict criteria for seeing long COVID patients as patients. They didn't have to be hospitalized. As a matter of fact, we actually leaned towards people who were not hospitalized, because those were people that were maybe not having as much attention as the hospitalized patients. So, and we also didn't require, as again, patients, we didn't require a positive test. But to be as rigorous as possible for research, anyone with long COVID that was enrolled into our JAX cohort was required to have a positive test. So that could be positive antigen test or positive PCR. It's also important to note that and it could be positive antibodies too. And this was before vaccines became widely used. So those 3 criteria were used to enroll our long COVID patients. Sick, they had to be sick with symptoms for at least 3 months and have a positive test. Do you want to add anything more to that, Derya?
No, I think that's great. Thank you.
Thank you for that insight. And how are you working to characterize whether long COVID patients are meeting the criteria for MECFS?
Well, again, that's a big question. MECFS is the prototypical post-infectious disease, right? So we saw long COVID as an opportunity to determine how many, whether or not people with long COVID actually progress to an MECFS diagnosis. Many of the patients that we enrolled in JAX were sick, got sick with COVID in March of 2020. And we started enrolling them in January of 2021. So the first people were sick in March 2020, but we actually have people with long COVID up until, boy, I guess it's going to be December of 2021 and in the beginning of December of 2020 and in the beginning of 2021. So that we have a spectrum of people. And again, by definition, for MECFS, you have to have had symptoms for 6 months or more. So many of our patients, our long COVID patients, have symptoms for 3 months or more. And as we follow them over time, we are seeing kind of the evolution of symptoms. We will see the evolution and natural history of their immune profile, et cetera. So our aim is not necessarily to see if they meet criteria, but to collect the information to see how they are similar or different from MECFS. And we will have the information, both the clinical information and the all of the immune and metabolomic and microbiome data to see how they compare to MECFS and whether or not, again, they're similar or different, and whether or not some meet MECFS criteria along the line, down the road.
Thank you so much. And our last question. So given what you have learned in MECFS and what is known about post-viral illnesses, what do you think would be useful for long COVID researchers to know, or what would you recommend researchers be looking at in terms of immune perturbations and microbiome disturbances?
So maybe I'll start with that, and Julia can add on the microbiome aspect of it. First of all, I mean, there's really no question in my mind that long COVID will end up having a very significant immune perturbation component. I mean, in fact, it's sort of watching MECFS-like disease happening in real time. As terrible as tragedy as this was, and it was also, as Suzanne pointed out, was an opportunity to really, catch what would start as an MECFS early on. It's conceivable that long COVID will be sort of a more severe aspect. Maybe other post-viral illnesses start kind of more milder and get worse over time. So we don't yet know that. But there's no question that there's going to be an immune component. There's also, I think, very likely that metabolism is going to be involved, because we know that during COVID, for example, if you're pre-diabetic, you become diabetic after COVID. We know that diabetics have much higher risk of, for example, dying from COVID. Obesity has a major risk factor. So they're all related to the metabolic dysfunctions, which COVID makes, the SARS-CoV-2 makes worse. I think there's some evidence, and Julia can respond to that. The microbiomes probably is going to be involved as well. Now, to answer specifically the question, it's conceivable that some of the immune profiles that we found earlier are going to be involved in long COVID. But I think we're in a very unique position, because we have this subgroup of patients who are less than 4 years of their MECFS. And again, it's most plausible to hypothesize that the long COVID patients, because they are still early on, they're going to look more similar to those less than 4 years of MECFS rather than the long term. So that data is still being processed. We know some of it from the metabolism microbiome. Maybe Julia, if you want to add a few words on that.
Yeah, so just to add, I think I agree with Derya that there's a tremendous number of potential parallels, and I would take the same approaches as we've taken here to look at how the microbiome might be contributing to predilection and severity of long COVID. So, one thing that I think I didn't mention earlier, but I just want to emphasize, is that there's been increasing number of publications now that indicate that the gut microbiome can predict the plasma metabolome, up to 15-20% of the chemicals basically floating around in our blood, and as well as vice versa. So this is a complex and very substantial bidirectional exchange between the microbiome and circulating metabolites, which then have numerous systemic effects. the diseases that are that increase the susceptibility and severity of COVID and likelihood of long COVID likely as well, like obesity. These metabolic diseases have also been increasingly linked with dysbiosis of the microbiome. And so I think that, you know, they're again emphasizing that there's going to be a lot of parallels here, and the early onset, I would say MECFS will probably be most analogous in this case.
Just to add one last thing related to that, I think there's also very good evidence that SARS-CoV-2 infects our intestines. In fact, you know, in a number of people, it can lead to a sort of a months-long, you know, semi-chronic infection. In fact, you know, in feces, you can find virus in large amounts, and people even measure PCR from the sewerage to determine the load of the virus. So I think, you know, clearly the virus is doing something in the intestine and perhaps causing some leaky syndrome, the microbiome leakage and so on. So this is going to be really fascinating to figure out, and I agree. Hopefully, the stuff that we're doing will be a sort of a template for the long COVID researchers.
Great. Well, thank you so much. And on behalf of the entire Community Advisory Committee for the NIH MECFS Research Network, I just want to thank you so much for taking the time to answer our questions. We learned a lot, and there was a lot of interesting continuing questions going on that we hope to see in future research. So thank you, the three of you, for coming and talking with us today. And also thank you to the whole Jackson Laboratory team for the work that you do.
Thank you as well.
Thank you.